Okay, welcome back, guys. We're going to launch into a new section on genetics and biotechnology. Up to this point, we've been differentiating among these little guys, these microbes, looking at shape, whether it's macroscopic or microscopic, the structure of the cells, the abilities, meaning the expression of enzymes that would allow for certain kinds of metabolism to occur. And now we're going to look at the underpinnings for a lot of those behaviors, those types of metabolism they're capable of, by looking at genetics. All right. Now, this is a pretty dense subject, and we do have antibiotics as part of this particular third module. So it's kind of a dense part of the course, but we are halfway through the course, so there's light at the end of the tunnel. And I think you'll find it really interesting because genetics are cool, and the antibiotic stuff will give you a real kind of clinical, medical insight as to how these antibiotics work. All right, so let's launch right into genetics here. We're in Chapter 8 of your text online, and we have a wide variation of genome size when you look at living things. At the top here in our list here, we've got humans with 10 times 10 to the 9 for the base pairs, the nucleotide pairings that are in the DNA, and then we have all the way down to 4 or 5 million base pairs when you have E. coli, and of course viruses can have a much smaller amount of genetic material because they depend on host cells to do most everything. But viruses like HIV virus is only, I think it's 9 genes total, okay? So one big thing though is that we need to differentiate between is we have now left a section where we're talking about abilities these cells had, the expression of genes which resulted in enzymes, okay? And that would show abilities, the expression, okay? The phenotype is going to show itself in what do the cells look like structurally, how do they behave, and that could entail the enzyme activity, okay, and metabolism. And now we're looking at the genetics. So phenotype is the expression of the genes and what that will look like structurally or enzymatically. And then we have genotype, which is under the underpinnings, the genetic material. All right. Now, a lot of this was in your chemistry review, so I know that a lot of you know this, so I'm not going to spend too much time on the basic structure of DNA. But DNA is 2 chains bound together by hydrogen bonds. Okay. Each nucleotide, which is of course the base unit or the smallest unit of DNA, actually has sugar, a base, and a phosphate group all bound into one. Okay, so that's a nucleotide, and the bases are what give each one of these nucleotides their unique look. Right? We have G, A, T, and C. These are our 4 different nucleotides when it comes to DNA, and those are the bases. These bases can bind to one another via 2 hydrogen bonds across those 2 chains, or they can bind to one another by 3 hydrogen bonds. But just like proteins, these hydrogen bonds are critical to the integrity of the molecule. So here you see how A is always bind to T's and how G is also always bind to C's. And you'll need to know that because there'll be some questions in the test that will ask you what kind of pairings we would have in the process of DNA replication, which we'll go over, and then in the process of producing a protein from a gene. All right. Now, what we do is we refer to the ends of these 2 strands. When the 2 sides of DNA bind together, we refer to 5' and 3' ends of each of those strands. So I always think of it this way, okay? So if we got my hands, 5 fingers on this side, 5 fingers on this side, there's a 5' end, okay? Where my elbow is, that would be the 3' end, okay? Now, what makes the 5' end 5 and what makes the 3' end 3? That has to do with the carbons in the actual sugar part of this particular nucleotide. There's a 5' carbon that is on the phosphate end, where it binds the next nucleotide, and there's a 3' carbon in the sugar on this other side. Okay, so we're talking about basically the carbons that are in the sugar. That's what gives us the 3' and 5' end. All right. All right, here's another image. I'm not going to expect you to reproduce these kind of chemical structures that you see here, but it's just kind of reiterating what I just said, right? We have a 3' end of the sugar that's able to bind to the next phosphate on the next nucleotide, okay? And then we have a 5' end that will bind to the nucleotide above that, right? And here again, it just shows you the A's and T's are together, C's and G's are together. We say that this is anti-parallel, like I say, right? They're going opposite directions. Let's see, what else would I want to tell you about this? Let's skip right on into how the DNA might replicate, right? So if we have two pieces of DNA, they make up one original DNA molecule. In order to replicate this so that growth can occur, right? We just got done talking about growth. In order for binary fission, for example, in a bacterium to take place, we need double of everything, including the genetic material, okay? So what will happen is these two pieces will be broken apart in some location, and you see this happening. Unwinding of the DNA has to occur, and then on each side of that, we're going to have a single strand that will need to be built upon to make our new strand, okay? So here we have a DNA polymerase, and it is acting upon this strand on the left. So we'll put this in red over here. Okay, so this strand over here on the left is a parent strand. So this would be one of the original strands for a cell that's going through replication. And then on that original strand, you're going to add on these various nucleotides, okay? And it makes sense that the polymer of nucleotides for laying down a new strand of DNA would be a DNA polymerase, okay? So one at a time, these nucleotides are brought in by DNA polymerase, and we have a brand new strand eventually at the end, okay? So here we show that happening with two original parent strands torn apart, okay? And then we build upon each of those strands to get daughter strands, all right? Again, DNA polymerase, that'll be the big one you need to remember, all right? Now this shows you really how complicated the process is. DNA replication involves three major phases or stages of the process. The initiation would have to involve breaking these two strands apart, the original parent strands, okay? So that double-stranded DNA is made into two single-stranded pieces of DNA, and then we have, like I say, that DNA polymerase coming on in and assigning complementary nucleotides, which basically means the bases are going to be paired up, all right, in the appropriate way. C's with G's, A's with T's, right? Now, here you see how awfully complicated it really is. There's a lot more enzymes than just DNA polymerase. However, DNA polymerase is the one that I'm going to ask you to know for sure, okay? And there are different kinds of DNA polymerase, okay, you can see here. But in general, all those DNA polymerase enzymes are basically bringing in each subsequent nucleotide and pairing them. Eventually, the termination of this would occur where the daughter strands have been formed and we have two separate double-stranded DNA again, right? Now, the fact that these words, initiation, elongation, where we just add nucleotides, and termination are found here does not mean that we're not going to find these phases again. We're going to see these phases 2 more times in this section. So if I were to ask you, what are the stages of DNA replication, and be able to explain what is occurring in those stages, you want to be able to do that and differentiate it from initiation, elongation, and termination when it comes to RNA formation and protein synthesis, which will come up later, okay? All right, so here we have what it might look like if we zoom out a little bit, okay? So here we have a parent strand, parent DNA here. We've got several locations where the DNA has been broken apart, right? There's 4 different locations in this particular image. And what we see on both sides is that DNA polymerase is going to work in both directions to eventually form 2 new daughter strands at the bottom here. So what these are called are origins of replication. In this case, we have 2 linear pieces of DNA, right? They don't connect up at the end. They go clear across, and they have 2 ends, right? So in this linear piece of DNA, we see many origins of replication. Now this is going to be different when we look at bacteria. We know bacteria don't start out with linear pieces like this, right? They start out with circular pieces. And in prokaryotes, bacteria, for example, there's only 1 origin of replication. So already we're seeing a difference here, and you'll want to be able to distinguish how DNA replication would be different in a cell of my own, a eukaryotic cell with linear chromosomes, and a prokaryotic cell. And the first one we see here, right? 1 origin of replication in bacteria, many in a linear chromosome like we see in a eukaryote. Right? Now either end of this replication bubble you see here, we're going to have, it's almost like it's a fork in the road, right? And those are called replication forks. Now why do we call these bubbles, these replication bubbles? Well, the first time when we saw these using electron microscopy, we had what looked like little bubbles form, okay, like you see here in the image that we would get from DNA when it was undergoing replication. Okay, so here we see a bubble, here we see another bubble, and now we know what's going on there, of course, right? Now here we see how it might look differently if we have the circular chromosome like you see up here for the chromosome of a bacterium, okay? Here you've got the circular form, and we have one place where we have an origin of replication. We see our bubble form, right? And then eventually that bubble just keeps getting bigger and bigger and bigger until like a magic trick, we got 2 rings that are coming apart, right? And those 2 rings, of course, are going to be our daughter DNA with the new strand on each one of them in pink, right? All right, the final slide for this clip is, and you'd want to be able to do this kind of thing on our exam, all right? If I pose to you this particular string of nucleotides, and I tell you that this is one side of our DNA molecule, right? So I'm suggesting to you that this particular sequence could easily be a sequence, and I'll just, we could just cut right out right here, right? In that particular molecule. And I'm going to ask for you from that blue strand there, okay, how is it that we would add nucleotides, and what would be the sequence? Okay, during DNA replication, what would we get from this parent strand, in other words? Okay, now first thing I see here is that we have actually a 5' and a 3' end again, right? It so happens that when DNA polymerase comes in here and starts building upon the parent strand, it is going to go always in a, it's going to build in a 5' to 3' direction. Okay? It's going to build in that direction. Now, if we think about that, right? So here we have an original strand, okay? This is 5', this is 3'. But we need to build the new strand in a 5' to 3' direction. We do not want to start on the original strand where it says 5'. We want to start where it says 3' because this is where we're going to have our first 5' end of the new daughter strand, okay? So if I give you this kind of sequence, I tell you what the 5' and 3' ends are. Where would you want to start? You would want to start on the 3' end of the original strand. So I give you this DNA template, this original strand. You would want to tell me that we're going to begin on this side where we see the 3' with a cytosine, right? And we want to add the guanine with a 5' end on that side, okay? So the first nucleotide to go down would be guanine. Why? Because C and G's, they go together, right? These are going to have hydrogen bonds between them, okay? Now, we go to the next one. A's bind with T's. Well, next we see an A, so we need a T on this side, right? A G, so we see a C on this side. And you kind of get the idea, right? Eventually, your complement strand, the base pairs, they're going to be bound through hydrogen bonds, will be the following, and it'll go, of course, in that direction, 5' to 3', always in that direction, okay? All right, so let's call it quits for replication for now, and we'll move on to some stuff with producing proteins next.